Building vocabulary. A mutation is a change in the genetic material of a cell. A point mutation is a gene mutation in which a single base pair in DNA has been changed. A frame shift mutation is a mutation that shifts the reading frame of the genetic message by inserting or deleting a nucleotide. The word mutation comes from the Latin word mutare, which means to change. Which types of point mutations typically cause the most significant changes? When cells make mistakes in copying their own DNA, the resulting variations are called mutations. Mutations are heritable changes in genetic information. Mutations can involve changes in the sequence of nucleotides in DNA or changes in the number or structure of chromosomes. Mutations that change a single base pair are point mutations. Point mutations usually involve a substitution in which one base is changed to a different base. Substitutions usually affect no more than a single amino acid and sometimes have no effect at all. Mutations that don't affect amino acid sequences are known as silent mutations. Mutations that change the amino acids specified by a codon can be more significant and are called missense mutations. If a mutation changes an mRNA codon to result in a stop codon, it is known as a nonsense mutation because it causes translations to stop before the protein is finished. This can result in the production of a defective protein. Mutations in which one base or many bases are inserted or removed from the DNA sequence are called insertions and deletions. Insertions and deletions are also called frame shift mutations because they shift the reading frame of a genetic message. Frame shift mutations can change every amino acid that follows the point of the mutation. They can alter a protein so much that it is unable to perform its normal functions. Chromosomal mutations involve changes in the number or structure of chromosomes. These mutations can change the location of genes on chromosomes and can even change the number of copies of some genes. There are 4 types of chromosomal mutations: deletion, duplication, inversion, and translocation. Deletion involves the loss of all or part of a chromosome. Duplication produces an extra copy of all or part of a chromosome, An inversion reverses the direction of parts of the chromosome. Translocation occurs when part of the chromosome breaks off and attaches to another. A mutagen is a chemical or physical agent in the environment that interacts with DNA and may cause a mutation. Polyploidy is a condition in which an organism has extra sets of chromosomes. The suffix gen means producing. How does a mutagen produce a mutation? Genetic material can be altered by natural events or by artificial means. The resulting mutations may or may not affect an organism. Some mutations that affect individual organisms can also affect a species or even an entire ecosystem. Many mutations are produced by errors in genetic processes. DNA replication results in an incorrect base roughly once in every 10 million bases, but small changes in genes can gradually accumulate over time. Some mutations arise from mutagens, chemical or physical agents in the environment. If these agents interact with DNA, they can produce mutations at high rates. Cells can sometimes repair the damage, but when they cannot, the DNA base sequence changes permanently. The effects of mutations on genes vary widely. Some have little or no effect. Some produce beneficial variations, and some negatively disrupt gene function. Many mutations are neutral. They have little or no effect on the expression of genes or the function of the proteins for which they code. Whether a mutation is negative or beneficial depends on how its DNA changes relative to the organism's situation. Mutations are often thought of as negative, since they can disrupt the normal function of genes. However, without mutations, organisms could not evolve. Mutations are the source of genetic variability in a species. Some of the most harmful mutations are those that dramatically change protein structure or gene activity. The defective proteins produced by these mutations can disrupt normal biological activities and result in genetic disorders. Some cancers, for example, are the product of mutations that cause the uncontrolled growth of cells. Mutations often produce proteins with new or altered functions that can be useful to organisms in different or changing environments. Plant and animal breeders often make use of good mutations. For example, when a complete set of chromosomes fails to separate during meiosis, the gametes that result may produce organisms with extra sets of chromosomes. The condition in which an organism has extra set of chromosomes is called polyploidy. Polyploidy plants are often larger and stronger than diploid plants. Important crops have been produced this way.